So, good morning to all of you, students of BT third sem. Today, I'm going to discuss with you about the important topic that is given in your molecular biology syllabus, which is your translation in prokaryotes and eukaryotes. Um, so, let's get started now. Now, it will be a very lengthy topic, so you have to watch it with patience and you have to study it carefully. Okay, if you have regarding any doubts after this lecture, you can ask me in the WhatsApp group. Okay, so let's get started now. So, about the translation process in prokaryotes and eukaryotes. Okay, so let's begin. So normally we all know that DNA is normally transcribed to form mRNA, and that mRNA is further processed. In case of eukaryotic cells, they have to do a processing of the mRNA, like your 5 prime cap formation, then polyadenylation at the 3 prime end, and splicing, where we removes the introns and we join the exons, which codes for the protein to form a mature mRNA, and that mRNA will be translated by the process of translation to form a linear sequence of proteins, which will be further modified by post translational modification to form a proper functionable protein. Okay, so this is the overview of your central dogma of your molecular biology, where we have done the transcription process. Well, the splicing process has not been discussed till now. We will discuss them soonly after this translation process, and then another video lecture I will provide to you maybe tomorrow or day after tomorrow, which is your the DNA damage and the DNA repair systems. Okay, then we will do the splicing process. So till now we will discuss about the translation. So what happened in translation? In the translation process, decoding of the nucleotide sequence into linear sequence of amino acids of the polypeptide chain occurs. So what happened in translation? The nucleotide sequence, which is your mRNA here, that nucleotide sequence will be decoded into linear sequence of amino acids of the polypeptide chain that will occur, right? So here the mRNA will be using as a template, and not as a template, it will be using for the synthesis of the proteins, for the synthesis of the amino acids, and these amino acids will be joined by the peptide bond. Okay, and then it forms a linear chain of amino acids, that a polypeptide chain will be occurring. Okay, so for the translation process to begins, many things are required, just like um, when you are cooking some of your favorite dishes, many components are required for that, many ingredients are needed. So just like that also in translation, many components are to be required, but the most important is four major things are there which has to be needed during the translation process. And what are those four things which are needed for translation process to begin? Without these four things, your translation will not begin. The first is your mRNA. Yes, without mRNA, you cannot able to prepare your polypeptide sequence. You cannot able to prepare your amino acids, the sequence of amino acids, which will encode for that mRNA. So what it is written here? From this mRNA, protein will be synthesized. So it's a very important thing. mRNA consists of series of codons. So it consists of series of codons. Codons are what? Codons are three nucleotide long decoding element. That means codon is made up of three nucleotides, here ribonucleotides, which code for a specific amino acid. And these codons are adjacent to each other. That means they are located next to each other to form a series of codons, what we call as open reading frame. Well, open reading frame is the series of codons that start with start codon, that is your AUG, and ends at stop codon, which is your UAG, UGA, and UAA. So this thing I have said in your genetic code lecture. Okay, so this they have this series of codons which are adjacent to each other, and this codon will code for that specific amino acid. Actually, what happened? This codon are recognized by your the tRNA. tRNA also have a specific sequence known as anticodon sequence. They have anticodon sequence. With the help of anticodon sequence, specific tRNA will come and interact with this coding sequence of mRNA. And tRNA also carries a specific amino acid respective to that codon sequence found in the mRNA. Right? Okay. So in this way, your mRNA is very important. Well, tRNA is also important, that we will see next in this after this mRNA part. Let us see. So I have said already this one, that codons are three nucleotide long unit, and each codon will code for a particular amino acid. Okay. So that's why mRNA is required. More information of mRNA will be provided after this page. Next is your tRNA. Well, T means transfer RNA. tRNA consists of anticodon sequence. So it has anticodon sequence. This anticodon sequence is specific and it is complementary to the coding sequence of the mRNA. So tRNA consists of anticodon sequence, and this anticodon sequence is complementary to the coding region of the mRNA. And this anticodon sequence, they base pair with the coding sequence with the help of a specific type of base pairing known as Ubel base pairing concept. Ubel base pairing concept. Well, this has not been mentioned in your genetic code. I will discuss them separately on next video. It actually tries to say that the third base in the codon, well, suppose AUG, okay, AUG code for methionine. So this is the first base, okay, I'm marking. This is the first base A, second base is U, and the third base is G. So the third base of the codon is not so much essential for coding particular amino acid, and due to which Ubel base pairing comes. Ubel base pairing occurs between the tRNA's anticodon with the mRNA coding region. You will see them in that video. Okay, so tRNA have anticodon sequence, and this anticodon sequence is complementary to the coding sequence of the mRNA. So the mRNA has multiple codons, as you know, and respect to that tRNA's anticodon sequence will base pair with that specific codon and will carries that specific amino acid for that particular codon. tRNA, how it carries the amino acid? tRNA have an acceptor arm. It has an acceptor arm which represents as 5 prime CCA 3 prime. This is the sequence of the tRNA acceptor arm, 5 prime CCA 3 prime, which is required for the which is required for the amino acid binding. Okay, so this acceptor arm is the site where your amino acid binds, and it is catalyzed by an enzyme, and the name of the enzyme is this amino acyl tRNA synthase. Amino acyl tRNA synthase is the enzyme which catalyzes this process of attaching amino acid with the tRNA in a two-step reaction. Well, you can see amino acyl tRNA synthase. This enzyme helps to bind a particular amino acid to the tRNA molecule. So it actually catalyzes the binding of specific amino acid to the tRNA's acceptor arm. So it attaches the tRNA in the acceptor arm, that amino acid, and it makes the tRNA then functional for the translation process. So when tRNA attach with specific amino acid by the help of this enzyme, it becomes what we called as amino acyl tRNAs. It becomes amino acyl tRNA. Now this amino acyl tRNA becomes functional. It now becomes functional for the translation process. It is carrying the amino acid, and it will be recruited to the ribosome by the ribosome, so that it can complementary base pair with the coding region of the mRNA by the oval base pairing concept, and then it allows that amino acid to be used for the protein synthesis. Okay, next is your ribosome. Well, ribosome, the entire translation process occurs within the large and small subunits of the ribosome, where the formation of the linear protein occurs. So, ribosome is the main site where this protein synthesis is being done, and it is done in large as well as in small ribosomal subunit. So assembly of the large and small ribosomal subunit is required, and here what I have drawn here, I have shown that the prokaryotic ribosomes, as stated here, prokaryotic ribosomes are called as 70s ribosome, whereas eukaryotic ribosomes are called as 80s. Well, these 50s and 30s are the large ribosomal subunit and small ribosomal subunit of the prokaryotic ribosomes, whereas 60s and 40s these are the large and the small ribosomal subunit of the eukaryotic 80s ribosome. Well, as you can see, the S has been mentioned here. S is actually what we called as Swedberg unit. Swedberg is a unit used to measure that how fast the molecules move in a centrifuge during centrifugation. So when we centrifuge, what happens? It rotates at a high speed, at a high rotation it moves, and during that time what happened, the ribosomes, their subunits get separated depend on how fast the molecules are moving in the centrifuge tube. So that's why prokaryotic ribosomes, their large ribosomal subunit are in case of prokaryotic ribosomes, their large ribosomal subunit is called 50s, and a small ribosomal subunit is called 30s, whereas eukaryotic ribosomes, their large ribosomal subunit is 60s, whereas small ribosomal subunit is 40s. Now, generally, bacteria, archaebacteria, they have smaller ribosomes and they are known as 70s ribosomes. And the 70s ribosomes are composed of small 30s subunit and a large 50s ribosomal subunit. So this was the overview of the important components required for the translation process to begin. Now let us see about the details of these of these components. So first is your mRNA. Well, mRNA, your mRNA, so the mRNA consists of a series of codons. So it contains series of codons, one codon right to next each other. They do not overlap. That means they don't overlap, but they are adjacent. They are located right next to each other. Suppose there is a AUG start codon, then there is a CUC, then there is GGG. So one codon right next one after another is present, and so these codons do not overlap. Okay, and codons are 3 nucleotide long unit, right? So it is made up of composed of 3 nucleotides only, and this series of codons make the open reading frame. Well, the open reading frame, or ORF, consists of series of codons which will code for a particular protein. So one open reading frame will code for a specific protein. An open reading frame begins from start codon and ends with stop codon. In between there are multiple codons, right, located one after another, coding for particular amino acid. In this way, open reading frame codes for a specific protein, and the whole translation process begins from the 5 prime end of the open reading frame. That means from the start codon of the open reading frame, your translation will begin, and it will proceed one codon at a time. That means the translation process will actually proceed one codon at a time. So it recruits one codon, allowing one codon, so that the specific tRNA carrying that amino acid for that particular codon enters to the ribosome. Then peptide bond formation occurs between the previously formed tRNA amino acid with the newly introduced tRNA containing that amino acid, and then the tRNAs are removed out. Okay, in this way, the whole translation process begin one codon at a time, and it is moving towards the 3 prime end, from 5 prime to the 3 prime end. Okay, so it's not so much fast as we see in replication or in transcription. 3 nucleotides, that means one codon is proceeded for the translation process. Okay, after attachment of the amino acids, then again ribosome will move for the next codon to incorporate next amino acid, to incorporate next amino acid tRNA into that codon. Okay, proper anticodon codon base pairing occurs, and then it will be recruited by the large ribosomal subunit to form to catalyze that peptide bond formation between the amino acids of respective tRNA. So the first and the last codon of your open reading frame is called the start codon and the stop codon. So where your open reading frame is initiating first, that is called a start codon, and the place where the open reading frame is ending, there that codon is called a stop codon. Start codon in prokaryotes, start codon is AUG and GUG, whereas in eukaryotes the start codon is always AUG. Well, why GUG is used as a start codon in prokaryotes? Well, there is some exceptional case. GUG normally codes for a different amino acid. GUG normally codes for a different amino acid, but when GUG is placed at the beginning of the open reading frame, that is at the first codon, if GUG is placed as a first codon of the open reading frame, then GUG code for different amino acid. Now this is an exceptional case because we know from the genetic code we have studied that one codon will code for one amino acid only, right? It does not code for different amino acid. Well, there may be more than one codons that code for the same amino acid, but one codon code for one amino acid. But here GUG has an exceptional case. It normally codes for a different amino acid. I think it codes for valine. It codes for valine amino acid. I think so. Just go and check. I have discussed this in your genetic code chapter, in genetic code lecture. But when GUG is placed at the beginning of the open reading frame, there GUG codes for methionine. In case of eukaryotes, they have only one start codon, which is your AUG. Well, stop codons are UAG, UGA, and UAA. Well, these codons do not code for any amino acid, but they will, after identification by the ribosomes of the stop codon, it will indicate a stop signal. It will indicate a stop signal which will recruits the other proteins which will terminate the translation process, like some proteins like RF1, also called as release factor 1, RF2, okay, release factor 2, then RRF, which is called as ribosome recycling factor. So these factors actually are then recruited when the ribosomes encounter this stop signal, when it observes, it identifies this stop signal. Then it recruits these factors called release factor 1, 2, ribosome recycling factor, so that the ribosomes can again goes and do the next round of translation in another mRNA. Okay, so stop codon do not code for any proteins. They do not code for any amino acids. Now, prokaryotic mRNA has more than one open reading frame. Uh, Prokaryotic mRNA has more than one open reading frame, so they are called as polycistronic mRNA. So it means that the prokaryotic mRNA will code for multiple proteins. This prokaryotic mRNA code for multiple proteins. Okay, multiple polypeptide chains will be prepared from prokaryotic mRNA, as it has more than one open reading frame. It has more than one open reading frame. That's why they are called as poly, means many, cistronic mRNA. Well, cistron means a segment of the gene that will code for a subunit of a particular protein or enzyme, whereas eukaryotic mRNA has only one open reading frame, so they are called as monocistronic mRNA. Eukaryotic mRNA has only one open reading frame that will code for one protein only. That's why eukaryotic mRNA are called as monocistronic mRNA. Why it is saying? Well, polycistronic mRNA refers to the messenger RNA or mRNA which encodes for two or more proteins. That means they have multiple open reading frame arranged in a linear fashion. So polycistronic mRNA are those mRNA which codes for more than one protein. That means they have multiple open reading frames. They have multiple open reading frames which are arranged in a linear fashion right next to each other. One open reading frame is there, then the stop codon is there, then again one again open reading frame is there. In this way, right next to each other, prokaryotic mRNA have multiple open reading frames. That's why we call the prokaryotic mRNA as polycistronic mRNA. Well, now let us see that how a picture has been given also about the polycystic and monocystic. We will see later on this picture on this video after this some pages. So what it is saying now is uh, during the initiation process of translation in prokaryotes, it involves a subunit of ribosome binding to a sequence on the mRNA known as Shine-Dalgarno sequence. So again I repeat, so for identification of the ribosomes to the mRNA in prokaryotic translation initiation process, so during prokaryotic translation initiation, the ribosome actually the small subunit of the ribosome, which is the small ribosomal subunit. The small ribosomal subunit is your 30S, right? It is your 30S. So the small ribosomal subunit or the subunit of the ribosome, it binds to the sequence on the mRNA, and that sequence where the mRNA where the sequence of the mRNA where the small ribosomal subunit bind is called Shine-Dalgarno sequence, or also called as ribosome binding site. In short, we call ribosome binding site as RBS. Okay, so initially ribosome attached to this ribosome binding site on the mRNA. Okay, now this is found in prokaryotic mRNA, the ribosome binding site, followed by rest of the ribosome assembly and translation machinery. Then it is followed by assembly of the other translation factors. They have also initiation factors, elongation factors. So initiation factors then assemble, that will then they will accommodate the large ribosomal subunit to bind and carries out the translation initiation. Okay, so there is a site called ribosome binding site, also known as the Shine-Dalgarno sequence, which is recognized by the small ribosomal subunit of the ribosome during prokaryotic translation initiation process in the mRNA. Then what it is saying, the ribosome then translate the protein and then falls off after elongation and termination. They will falls off from the mRNA and will translate the next round of translation. It will begin by binding to the mRNA. Now, in prokaryotes, there are multiple Shine-Dalgarno sequence, because usually we know that prokaryotes are polycystronic mRNA. They have multiple open reading frame. So, as they have multiple open reading frames, so there must be multiple Shine-Dalgarno sequence, or you can say they have multiple ribosome binding site that are present in each gene in the mRNA. This allows the ribosomes to assemble throughout the mRNA at each of the genes. So, as prokaryotic mRNA are polycystronic, they have multiple open reading frame, and just before to the open reading frame there is a region in the mRNA where the small ribosomal subunit interact and bind. That region is called, that region of the mRNA which is found in prokaryotes is your Shine-Dalgarno sequence, which specifically binds with the ribosome, specifically bind with the smaller subunit of the ribosome. Okay, and this Shine-Dalgarno sequence, what we call as ribosome binding site also, there are multiple ribosome binding sites actually. Um, because of having multiple open reading frame, they have also multiple Shine-Dalgarno sequence. Thus it allows the ribosomes to assemble throughout the mRNA at each of the gene. So for each gene there is open for each gene there is open reading frame, and ribosomes assemble just before the open reading frame with that ribosome binding site. Okay. Now, eukaryotic mRNA are monocystronic. That means each gene code for only for a single protein and has only one initiation and one termination codon. So eukaryotic mRNA are monocystronic. They have only one open reading frame. Their gene will code for only one protein and has only one initiation. That means the start codon and one termination codon. What's the reason behind that? Eukaryotic genes are not present continuously. That's the main reason. Eukaryotic genes are not present continuously. The exons or the coding region are interspaced with the introns, which are the non-coding regions. So, if you see the eukaryotic mRNA, they have introns and exons. They have introns and exons interspaced with one another. So in order to, as the eukaryotic genes are not continuous, as the eukaryotic genes are not present continuously, the exons are interspersed with the interspaced with the introns. So we have to remove these sorry, not exons, we have to remove these introns. We have to remove these introns. We have to remove these introns and ligate these exons to make a functional mRNA. And that's why, due to this processing process, eukaryotic genes have only they are not continuous. Okay. Second thing, reason behind the monocystronic mRNA in eukaryotes is that eukaryotic system is a very complex system which needs regulation at many levels for proper expression of the gene. So, unlike prokaryotic regulation, they are regulated by simple ways like tryptophan. You can see that operon concept I have discussed with you before, but in eukaryotes they have a more complicated system, so their level of regulation is very high. So they needs regulation at many levels. Actually at many levels meet at many levels, it means they have to regulate at replication level, they have to regulate the gene expression at transcription level, at translation level. At each level, gene regulation of eukaryotes are carried out in eukaryotes is carried out. So that's why eukaryotes have only are monocystronic. They have only one open reading frame that will code for a particular protein. Then another open reading frame will be present somewhere else. So the open reading frames are not continuous in eukaryotic mRNA. In polycystronic genome, all the structural genes of an operon are turned on or off together due to the presence of a single promoter and operator upstream to them. So, as we have seen in our operon concept, when we have studied about the operon, what we have seen that in polycystronic genome, those who are polycystronic like prokaryotes, all the structural genes of an operon are turned on or off by the by the repressor protein, right? When they bind to the operator, they cause the operon to turn on and to turn off. In this way, they regulate the genes which will code for the proteins. Okay, and they have a single promoter. Okay, polycystronic genome have a single promoter where RNA polymers will allow to do the transcription process, bind and do the carry out the transcription process, and operator which is located upstream to the genome. Okay, gene. But in case of eukaryotes, many cases one single protein is involved in many processes. So eukaryotes have to evolve to have monocystronic genomes. Now, this is different from eukaryotes. Polycystronic genomes, they have one promoter, one operator, and all the structural genes. They also have a repressor region where repressor protein will be prepared and will allow this regulation of these genes in polycystronic genome. But in case of eukaryotes, a single protein, in case of eukaryotic genomes, a single protein will be involved in many processes. That's why that's why the concept of exon shuffling comes. In exon shuffling, all the exons are not arranged in a proper manner. There may be shuffling occurred among the exons of a particular mRNA, which will code for different protein. So a single protein, in case of eukaryotes, a single protein may be involved in multiple processes. So eukaryotes has to be evolved to have monocystronic genomes. So these are the main causes that why eukaryotes have why eukaryotic mRNA are monocystronic. The first point that they have mentioned, that eukaryotic genes are not present continuously, because they have intron regions, which are the non-coding regions, interspaced with the exons, which are the coding regions. So in the introns has to be removed out. In the third point, they have also mentioned that in the third point they have mentioned that in many cases the one single protein is involved in many process. So how it is possible? Suppose there is mRNA, eukaryotic mRNA, where multiple introns, exons are there. So you can see I am writing intron, exon. Okay, I am writing intron, exon, intron. Okay, and I am naming that intron 1, intron 2, intron 3, intron 4, exon 3, exon 2, exon 1. So during splicing process, obviously these exons will be joined because they code for they are the coding regions. They are the coding regions which will code for a particular protein. Okay, during translation, E1, E2, E3. But what happened, one term what we call as exon shuffling also occurs. It may occur like this also. It may occur like E1 and E3 have combined. Okay, during the splicing process. So it will code for a different protein. Okay, then it may be possible that E2 combined with E3, or suppose the arrangement becomes different, like for example, in this way it can occur E3, E1, E2. So different pattern of exons have been joined to form a to make the protein, but they will now have a different function. So this is called as exon shuffling. So due to this reason, that a single protein one single protein may involve in many processes. That's why eukaryotic mRNA are usually monocystronic, and this is the main reason. But in polycystronic genome, which are the prokaryotic genome, they have all the structural genes of a operon right next to each other, and they are turned on and off by the regulation of the operator, promoter, and the repressor gene. As we have seen in the operon, that how a repressor protein is regulating the operator to turn on and off, and allowing the RNA polymerase to regulate the transcription of all the structural genes together. But in case of eukaryotes, it does not occur because one protein can be involved for many functions. One reason I have shown you the exon shuffling, where the exons are shuffled, they are mixed in different combinations to make a functional protein having different function. Okay, now it is saying that upstream and downstream both refers to the relative positions of the genetic code in the DNA and RNA. Here, what is upstream and what is downstream, I have written here. So the upstream and downstream refers to the positions or locations of the genetic code both in the DNA and RNA. When we are talking about upstream, we have to look towards the 5 prime end of the mRNA molecule, and if you are talking about downstream, you have to look towards the 3 prime end of the molecule, the mRNA molecule. Promoters, operators, even the ribosome binding site, they are also located to on the upstream. Okay. Now I will talk you about the ribosome binding site, which I have just overview I have said in this in before just little bit ago. So prokaryotic mRNA have a ribosome binding site, or RBS. It is also called as signed Dalgarno sequence after the name of the scientists who have discovered this region. For the translation process to occur, ribosome must bind to the mRNA, right? So the ribosomes are the sites of protein synthesis, and mRNA is also needed for the protein synthesis, because the mRNA contain the codons which will code for the amino acid. So we need ribosomes to be attached to the mRNA. And how it is possible? It is possible by the ribosome binding site, which is found in the mRNA in prokaryotes. So ribosome binding site, also called as signed Dalgarno sequence, it is typically located 3 to 9 base pair upstream, that is on the 5 prime site of the start codon. So if you want to see the location of the ribosome binding site in prokaryotic mRNA, ribosome binding site is located on the upstream, that means it is located towards the 5 prime end. 3 to 9 base pair upstream, that is on the 5 prime site of the start codon. So it means to say that this ribosome binding site is located 3 to 9 base pair upstream from the start codon, where the translation process will begin. So at the beginning of the translation is your start codon. From the start codon, 3 to 9 base pair from the start codon, there is a ribosome binding site towards the 5 prime end. Towards the 5 prime end, there is a ribosome binding site located 3 to 9 base pair upstream from the start codon. Okay. Now, this sequence of the ribosome binding site, why ribosome binding site allows the small ribosomal subunit to bind, because the sequence of ribosome binding site is complementary to the 16S rRNA. 16S rRNA is a component of the small ribosomal subunit. So the small ribosomal subunit has many components, and one of the component of the small ribosomal subunit is your 16S rRNA. This 16S rRNA, the sequence which is found on 16S rRNA is complementary with the ribosome binding site in the mRNA. That's why the small ribosomal subunit able to bind with the mRNA at this ribosome binding site. So the ribosome binding site can base pair with the 16S rRNA, thereby beginning the translation process. This is the sequence of the ribosome binding site, as we can see here, whereas this is the sequence of the 16S rRNA. Well, you can see they are quite complementary with each other. That's why the and 16S rRNA is actually the component of the small ribosomal subunit. So easily the small ribosomal subunit identify this ribosome binding site and binds. Base pairing occurs and binds very easily. Okay. Okay. So this is actually about what I have tried to draw about the polycistronic mRNA, which is found in prokaryotes. We can see two open reading frame here, one located here, another located here. Now you can see the start codon and the stop codon, here also a start codon and a stop codon. Just before the open reading frame, there is ribosome binding site. Just before the open reading frame, there is a ribosome binding site which is located 3 to 9 base pair upstream from the 5 prime side of the start codon. Okay, so you can see here two ribosome binding sites are there. So here two ribosome binding sites are there. So multiple ribosome binding site is present where your ribosome can able to interact with this ribosome binding site and catalyze the attachment of its small ribosomal subunit with the mRNA. Okay, so multiple ribosome can bind here, one ribosome here, one ribosome here, and carries out the translation process in each of these open reading frame. Okay, now about the eukaryotes, if I say eukaryotes don't have this ribosome binding site. Okay, so eukaryotic mRNA have 7-methyl guanosine triphosphate cap at the 5 prime end and a poly A tail at the 3 prime end. So if you see the eukaryotic mRNA have a cap at the 5 prime end, and the name of this cap is 7-methyl guanosine triphosphate cap. 7-methyl guanosine triphosphate cap, the name of that cap located at the 5 prime end. Now this cap, and there is a tail also in eukaryotic mRNA at the 3 prime end known as poly A tail at the 3 prime end. So this 5 prime cap is catalyzed by three enzymes. As we have seen in post transcriptional modification, we have seen that this 5 prime cap is catalyzed by three enzymes. Like the name of these three enzymes is RNA triphosphatase, guanylyl transferase, and methyl transferase. So three enzymes are responsible for the cap formation in the 5 prime side of eukaryotic mRNA, as we have seen in RNA processing. And the poly A tail is actually catalyzed by polyadenylation enzymes. So some polyadenylation enzymes are involved for the formation of the tail, a polyadenylate tail, like CPSF, like CSTF. Okay, these enzymes recognize the poly A signal found in the DNA and allow the poly A polymerase for addition of adenine residue at the 3 prime end. Well, CPSF is your cleavage and polyadenylation specificity factor, written right here, whereas CSTF is cleavage stimulation factor. So ribosome normally identify this 5 prime cap. Well, the polyadenylate tail is actually recognized by the translation initial factors. So the initial translation factors recognize the 3 prime tail of the eukaryotic mRNA, whereas ribosome itself recognize the 5 prime cap and then binds to the mRNA. Well, the small ribosomal subunit actually recognize this cap and then it binds with the mRNA. Okay. Now, after bound to the mRNA, the ribosome starts to move. So when ribosome identify the 5 prime cap of the mRNA in eukaryotes, ribosome then bind and starts to move towards the 3 prime direction until it reaches to the start codon. This process is called as scanning. So when ribosome attach with the cap, the ribosome starts moving from 5 prime to 3 prime direction until it identifies a start codon, and this process is called your scanning. Okay, so this is how a simplified diagram of your eukaryotic mRNA looks like. Well, eukaryotic mRNA is called monocystronic. Why it is called monocystronic? As you can see, it has only one open reading frame that begins from start codon and ends with stop codon. Just upstream of this, at the extreme 5 prime end of eukaryotic mRNA, there is a cap. As you can see, there is a 5 prime cap here. At the 3 prime end, there is a poly A tail. This cap is actually recognized by the ribosome. The small subunit of ribosome recognize this cap, while the poly A tail is recognized by the translation factors called initial translation factors or initiation translation factors. Uh, there is various names. I will like EIF4F, EIF4G, different type of names of translation initiation factors are there, which recognize this tail, the 3 prime polyadenylate tail of RNA of your mRNA in eukaryotes, whereas the cap is recognized by the small ribosomal subunit, which allows binding of the ribosome to the 5 prime site and starts moving in 3 prime direction until it recognizes the start codon. This process is called as scanning. Now it has been observed that in some mRNA there is a presence of a purine 3 bases upstream from the start codon, and there is a guanine at the immediate downstream that will make a sequence called Kozak sequence look like this. So it means to say that in some cases there is a presence of purine 3 bases upstream from the start codon. So if this is the start codon, from here the open reading frame starts 3 bases, that is 1 2 3, after 3 bases towards the upstream there is a presence of a purine, either adenine or guanine, and presence of a guanine at immediate downstream, that means just after the start codon there is a guanine. So presence of this sequence called as Kozak sequence, and I think you have able to understood. I'm again repeating. It is saying that from the start codon towards the 5 prime end, that is towards the upstream, there is a presence of a purine, 3 nucleotides above, 3 nucleotides before the start codon. There is a purine at the upstream, and there is a guanine immediately after the start codon. This makes a sequence what we called as Kozak sequence, Kozak, Kozak sequence. And this sequence, they are talking that this sequence actually increases the efficiency of your translation process. So the Kozak sequence actually increase the efficiency of translation. It has been observed. Okay, what it was written, let us see. So it is written here that most eukaryotic mRNA lack this Kozak sequence. Most of the eukaryotic mRNA don't have this Kozak sequence, but the presence of this sequence will increase the efficiency of your translation process. So This sequence, which I am talking about, presence of a purine, which is located on the upstream, three nucleotides upstream from the start codon, and presence of a guanine at immediate downstream. That means next, right after the start codon, there is a guanine, makes a sequence called as Kozak sequence, and Kozak sequence increases the efficiency of your translation process. This is how your prokaryotic mRNA looks like. Look, so much, uh, so much so different open reading frames are there. You can see here a start and stop side. This is one open reading frame, this is another open reading frame, this is another open reading frame, and right at the upstream of the open reading frame there is a ribosome binding site, which is located right here. You can see here, in this ribosome binding site, the small ribosomal subunit will bind and initiate your translation process. Well, the small ribosomal subunit have a RNA called 16S rRNA, and the sequence which is found on the 16S rRNA is complementary with the ribosome binding site. That's why the small ribosomal subunit can bind to this ribosome binding site. They can bind to this ribosome binding site. Whereas in the eukaryotic mRNA, as you can see, eukaryotic mRNA has a 5 prime cap, which is recognized by the ribosome, the small subunit of the ribosome, and a polyadenylate tail, which is actually recognized by the initiation factors of translation like EIF4F, EIF4G. Okay, and here it has been shown that at the beginning of the start codon, uh, three nucleotides above the start codon, that means three nucleotide upstream of the start codon, there is a presence of a purine, either G or A, and a guanine immediate after the start codon. Okay, makes a sequence what we call as Kozak sequence. Kozak sequence. Okay, and this Kozak sequence actually increases the efficiency of the translation process. Okay, so this was all about the mRNA. Next, we will talk about tRNA. Well, we have studied many things about the tRNA structure. Still, we have to do this tRNA structure again, and we will proceed them now. So let's begin about the tRNA. Well, tRNA also called as transfer RNA. Transfer RNA or tRNA are the molecules that act as temporary carriers of the amino acid. So they carry the amino acid, uh, and specifically it carries the amino acid by matching the codon sequence of the mRNA with the anticodon sequence of the tRNA. So complementary base pairing, what we call as a type of base pairing called as Ubbel base pairing, occurs between the mRNA and the anticodon region of the tRNA, that will allow the tRNA to carry specific amino acid for that particular codon observed in the mRNA. So they means here tRNA bring the they may bring the appropriate amino acid to the ribosome based on the mRNA nucleotide sequence. So normally tRNA brings appropriate amino acid to the ribosome based on the mRNA nucleotide sequence. Okay, so based on the mRNA nucleotide sequence, tRNA bring the appropriate amino acid to the ribosome. Now it is also saying here tRNAs are also capable of forming hydrogen bonds with the mRNA. tRNA are also capable of forming hydrogen bonds with the mRNA. Additionally, this tRNA can also form ester linkages with the amino acid. Okay, again I repeat, tRNA capable to form hydrogen bonds with the mRNA. Okay, and additionally, this tRNA can also form ester linkages. They can form ester linkages with the amino acid at the 3' end of the tRNA, and this can be done with the help of the enzyme called amino acyl tRNA synthetase. So the ester linkage is that linkage which will attach the amino acid with the tRNA. I have written amino acid as this way, amino acid, so don't get confused. For a shortcut I have written in this way. So tRNA can attach with the amino acid with the help of ester linkage. So AA represents the amino acid here. Okay. So with the help of these ester linkages, tRNA is attaching with the amino acid, and how this is catalyzed by the help of the enzyme called amino acyl tRNA synthetase. Thus, tRNA can physically bring mRNA and amino acid together during the process of translation. So, it is acting as a coordinator. It is coordinating the attachment of amino acid so that it can carry that amino acid by observing the respective codon region of the mRNA. So, with the help of amino acyl tRNA synthetase, your tRNA uh, form linkages with the amino acid at their 3' end. So, at the 3' end of the tRNA, there is a acceptor arm. With the help of this acceptor arm, tRNA attach with the amino acids. Okay, and it forms a linkage actually, which is called as ester linkage, which is catalyzed by this enzyme called amino acyl tRNA synthetase. Now, tRNA pair with the mRNA in a complementary and anti-parallel manner. Okay, so generally this tRNA, they pair with the mRNA in a complementary and anti-parallel manner, and each tRNA can base pair with a stretch of 3 nucleotides on the mRNA. So the 3 nucleotides which is found on the mRNA, which we represent them as codons, they are base pairing with the tRNA. tRNA also have 3 nucleotide sequence called anticodons, where base pairing with the codon anticodon sequence occur in an complementary and anti-parallel fashion. Now these three sets of nucleotides on the mRNA called as codons and the corresponding sequence on the tRNA called as anticodons, what I have said. Okay, so with the help of the anticodon sequence found in tRNA, they complementary base pair with the codons in the mRNA and are carrying that particular amino acid respective to that particular codons. Now, base pairing between codon and anticodon brings specificity to the process of translation. So, how does base pairing of the codon and anticodon occurs? Base pairing between codon and anticodon occurs by the process of Ubel hypothesis, which was also proposed by the Watson have and Crick have proposed this concept, that how the third base of the codon is not so much specific for the particular amino acid, and how multiple tRNA can interact with the codons during the translation process to bring specific amino acid for the particular codon. So you will see them when I will discuss about the oval hypothesis in a different video. Okay. Now each tRNA will bind to a particular amino acid, and each will recognize a particular set of codon or codons in the mRNA. Most tRNA recognize more than one codon. It's due to the oval hypothesis that made them to do so. Because I have just now said, I have given you a hint that the third base of the codon is not so much specific to code for a particular amino acid. That's why most tRNA can recognize more than one codon, because remember, there are 64 codons, but there are 20 tRNA which will carry the particular amino acid. So there will be multiple tRNA which will identify multiple codons to code for particular amino acid. Okay, and this can be possible by the Hubble base pairing concept. Well, this you have also learned before when I was giving a video lecture on tRNA, that tRNA molecules are between 75 to 95 ribonucleotides in length, and all the tRNA have some common features, like at the 3' end tRNA have a sequence known as CCA sequence. This is called as acceptor arm, located at the 3' extreme end of the tRNA. Within this CCA site, CCA is what? It's the nucleotide sequence, the ribonucleotide sequence in the tRNA at the located at the extreme 3' end. So in this CCA site, the tRNA will bind to the amino acid with the help of the enzyme called amino acyl tRNA synthetase. So within this 3' end of the tRNA, which is called as acceptor arm, the tRNA will bind to the specific amino acid at the 3' end by the help of this enzyme called amino acyl tRNA synthetase. There are presence of some unusual bases in the tRNA as well. Some unusual bases are also present. These bases, these nitrogenous bases are not required for tRNA to function, but the cell who lack these bases, those cells, those who don't have these modified bases, which is found on the tRNA, will show reduced rate of growth. So I have said that tRNA have unusual bases as well. Some modified bases are also observed. Well, these bases are not required for the tRNA to function, but the cells who lack these bases, those cells who don't have this modified unusual bases, they show reduced rate of growth. So this suggests that these modified bases leads to the improved tRNA to function. So two important things we have seen here: tRNA have a CCA sequence at the 3' end. This within this CCA sequence, your tRNA will attach to specific amino acid by the help of your ester linkages catalyzed by this enzyme called amino acyl tRNA synthetase. Now, that CCS sequence which I am talking about is called acceptor arm also, and we have seen that there are some unusual bases, modified bases found on the tRNA. uh, those cells who don't have these modified bases, they show reduced rate of growth, and the efficiency of tRNA will become little bit reduced. So it suggests that this modified bases leads to improved tRNA 2 function. Now, what are the unusual bases which are found on the tRNA? So, what are the unusual bases which are found on the tRNA? D loop, T psi U loop, anticodon loop, variable loop, and acceptor arm. Well, unusual bases are not this. The unusual bases are named as dihydrouridine, pseudouridine. I am naming this here. Dihydrouridine, then pseudouridine, dihydrouridine. Then other bases are also there, which I am not naming here. So some these are the some unusual bases which are found on tRNA. And tRNA have certain loops. They have certain loop like structure, and they actually occur due to the interhydrogen bonding between them. They fold onto itself and form the loop like structure. And the loops which are found on the tRNA are your D loop, T psi U loop, anticodon loop, variable loop, and acceptor arm, which I have just mentioned a little bit ago. Well, D loop is called as dihydrouridine. D loop contains dihydrouridine, which is an unusual base. It is an unusual base which is found on the tRNA. Dihydrouridine is derived from your uridine by enzymatic reduction of the double bonds between the carbon atom at positions 5 and 6. So the dihydrouridine is a unusual base found on the D loop. So within the D loop there is a dihydrouridine. This dihydrouridine derived from uridine, and it can be derived from uridine by enzymatic reduction of a double bond that is located between the carbon atoms at position 5 and 6 of uridine. Okay. So this D loop consists of dihydrouridine, and the D loop has about 4 base pairs in length. D loop is widely believed that it acts as a recognition site for the amino acyl tRNA synthetase. D loop is widely believed that this D loop acting as a recognition site for the amino acyl tRNA synthetase, which is an enzyme involved in attachment of amino acid to the tRNA. This D loop also stabilizes the tRNA structure as well. This D loop also helps in stabilization of your tRNA. And this D loop allows as a recognition site for the amino acyl tRNA synthetase. That means amino acyl tRNA synthetase recognize this D loop. It will recognize this D loop, and will allow this enzyme amino acid tRNA synthesis. After identification of the D loop, it will attach to the 3' end of the tRNA and carry out that amino acylation of tRNA, where you will see the amino acid will be attached to the tRNA at the 3' end. D loop also stabilize the tRNA structure. Second loop that you will observe, you have seen also, is your T psi U loop. Well, the psi symbol that is given is referred to pseudo uridine. Psi refers to the pseudo-uridine. This pseudo-uridine is also derived from uridine, just like the dihydrouridine is derived from uridine by reduction of your double bonds that was located in the uridine between the carbon atoms 5 and 6. T U loop consists of 7 unpaired bases. That's why the loop forms, because no base pairing occurs. That's why loop like structure observed. So it has 7 unpaired base, and this T psi loop is very important because it serves as a ribosome attachment region. So this T psi U loop serves as ribosome attachment region. It will help to attach the ribosomes with the ribosomes during translation elongation. Next comes the anticodon loop, which is very important for base pairing with the specific codons in the mRNA. Anticodon loop consists of anticodon sequence which are complementary to the codons of the mRNA. Codons are also 3 nucleotides. Now, this anticodon region of the tRNA is a sequence of 3 bases that are complementary to the codon region of mRNA. Thus, they can bind to one or more codons for a amino acid. So, it means to say here this anticodon region of the tRNA is made up of 3 nucleotide bases which are complementary to the coding region of mRNA. Thus, they can bind to one or more codons for a amino acid. Well, these are the some unusual bases that I have said. Well, this is your uridine, and let us see in the D loop we have found a dihydrouridine. Well, I have said that when dihydrouridine is derived from uridine, and it occurs due to reduction by enzymatic reduction of a double bond between the carbon atom 5 and 6. Look, in the uridine, in the region 5 and 6, there is a double bond. This has been reduced, you can see right here, it has been broken off, and it leads to the form of dihydrouridine. Well, the same uridine can also mix pseudouridine. Well, pseudouridine is named because here you can see in pseudouridine what changes occur. The attachment site for the ribose sugar in uridine has been displaced, has been placed in different position. At the fifth position, the ribose sugar is being attached. So thus, it makes a different sense of uridine known as pseudo-uridine. Pseudo means inactive actually. The term pseudo means or dormant. You can better say dormant. Okay. So these are some unusual bases which are found on the tRNA. Well, now the variable loop. Well, variable loop is located between the anticodon loop and the pseudo-uridine loop. Variable loop will vary. It varies from 3 to 21 nucleotides. That means sometimes it is made up of 21 nucleotides, sometimes it is made up of 3 nucleotides, depending on which amino acid the tRNA encoding. So depending on what type of amino acid the tRNA is encoding, variable loop varies in their loop structure. Sometimes the variable loop is made up of 3 nucleotides only, or sometimes that cannot be seen at the time. Sometimes the variable loop is made up of 21 nucleotides. So depending on what amino acid the tRNA bind, variable loop also varies in their structure. Normally, this variable loop is very important in recognition of the amino acid synthetase for the tRNA. This variable loop also plays an important role in recognizing the amino acid tRNA synthetase for the tRNA. So, just like D loop, recognition is done by the variable loop for amino acid tRNA synthetase to carry out amino acylation of the tRNA. And just like D loop also, it also helps in stability of the tRNA. Last is the acceptor arm. Acceptor arm, as we have said before, it consists of 5' CCA 3' sequence, and normally this acceptor arm is needed for amino acid binding catalyzed by amino acid tRNA synthetase. So acceptor arm have a region called 5' CCA 3'. This region is very important for attachment of amino acid by amino acid tRNA synthetase. Well, up to this much, I have finished my lecture. It is to be continued in the next lecture. So, from the clover leaf model of tRNA, I will now again discuss with you in the next lecture. So, uh, thank you for all uh, to listen to my videos. Okay, and if you have any doubts regarding this lecture, you can ask me in the WhatsApp group. Okay, so just after little bit later, I will now start another lecture for the translation process. Okay.